Hi everyone, I hope I'm audible and welcome to today's week 10 live interaction session about biogeography and macroecology. So here we have Dr. Umesh Srinivasan and Dr. Vivi Robin. So we have got some questions in the discussion forum and I have shared the questions and yeah, students can ask the questions in the live chat, or they can unmute and ask. I think we'll start with the questions. If any of you have any questions, can start. Otherwise, we'll go to the discussion forum questions.
Sure. I think Robin has not yet joined. There are some
Yeah,
questions for him in the discussion forum. But what I do is the questions that are addressed to me, should I just
yeah,
start off with those?
sure. Yeah, you can.
Okay, great. All right. So, questions. This questions from Ram, which is that the slides have the logo, which frequently masks critical information. In this case, the author's name is masked. Yes, unfortunately, sometimes that happens where the logo masks the information. Let me just paste the author and the reference in the chat here. Um, So that's by Kevin Gaston. That paper is by Kevin Gaston in 2000 in the journal Nature. The next question is, how is wing loading measured? Wing loading is taking the weight of the bird and dividing that value, or taking the body mass of the bird and dividing that value by wing area. So that's how wing loading is measured. It's I believe that's how they do it in the aircraft industry as well. It's a measure of, it's sort of a proxy measure for flight ability, where species with different wing loadings are able to, migrate to different distances or disperse to different distances. It's sort of a proxy in birds for dispersal ability. That's how wing loading is measured. Um, so if you have any follow-up questions, please put those in the chat box. There is a question from Mayank, which is, when we're talking about competition and predation, and how competition and predation um, uh, might vary across latitude, for example. So this is a question about the latitudinal species diversity gradient, and that gradient is in species which is that the tropics have more species than higher latitudes. So as you go towards the poles, the number of species keeps coming down. And there's more than 100 explanations for the latitudinal diversity gradient, many which are not mutually exclusive. They can all be operating together to create the latitudinal diversity gradient. Mine, as an observation, that some of the theories propounded for the latitude diversity gradient seems to be the effect of the latitudinal diversity gradient itself. For example, the presence of more predation and competition can be the result of there being more species, right? So more predation and competition in the tropics, maybe that's just an outcome of the fact that there are more species in the tropics. Similarly, the presence of a larger number of niches in the tropics can also be because of higher species richness in the area. So, and he says, can you comment on this? Absolutely spot on observation. That's absolutely correct. Um, so, almost all the explanations for the species richness gradient with latitude are observational, right? It's in some cases like predation, competition, number of niches, and so on. It's very difficult to separate what is the cause and what is the effect. To do that, what you need to do is experimentation. And experimentation on such large scales is, for all practical purposes, just impossible to do that. Right? It's um, to actually alter the number of niches across a latitudinal gradient, that might not be easy to do, and so a large number of the inferences on the latitudinal diversity gradient are the result of examining patterns and correlations. For example, correlations with temperature and species richness, correlations between rainfall and species richness. Um, but absolutely, there could be a lot of interplay between these factors. A lot of these factors might be reinforcing each other. So a large number of species could lead to greater number of niches. The greater number of niches then are involved in maintaining that greater number of species in the tropics. So yes, it's, for all practical purposes, very, very difficult, if not almost impossible, to fully understand why there are latitudinal diversity gradients and what is the precise interplay of those factors leading to that latitudinal diversity gradient. There is a second question from Mayank, which is: with the past few slides, we can deduct that range size, the geographical range size of a species measured, for example, in square kilometers, is inversely proportional to species richness. Or in other words, as we move away from the equator, as the species richness reduces, range size increases. Yes, that is true. When you move away from the equator, you have a low number of species, but each of those species, on average, occupies a larger geographical area. The species range itself is larger at the equator, on average. Uh, is larger at the equator, is larger at the poles than it is at the equator. So there is an inverse relationship between species richness and range size. That is true. So Amrita, I think those are the only questions on the discussion forum.
No, I think that's all about the questions regarding the macroecology. All those questions are for biogeography, rest of the questions. I think, yeah, you answered almost all questions. There is no question, yeah.
Yeah, so are we waiting for Robin?
Yeah. If anybody have any questions, please.
Mr. Ramurthy is pointing out that Arctic wren must be the best example for range. So the Arctic wren, the winter wren, is a species that actually has a very, very large range. It's found in the higher latitudes. It's found uh, uh, in temperate regions. And yes, it's got a very, very large range. It's a classic example of a high latitude species that also has a very large geographical range.
Hi, Robin. Uh,
I'm trying to look at any questions that are there.
so I have shared a Google Doc with questions from discussion forum. So I can read out the questions for you. Is that okay?
Yeah, please.
So there is a question from Ram. Are Galapagos finches and Hawaiian honeycreepers are relicts? So that's the question.
Are,
Hello?
are they relicts?
Yeah,
Hello? Yeah.
yeah,
Like, are they relicts? The question is if they are relicts, is it?
yeah. Are Galapagos finches and Hawaiian honeycreepers relicts? So you mentioned about the relicts. Um,
Uh, I'm not sure if I remember the context of this. But usually relics are species that have stayed on while the surrounding other species have kind of gone extinct. So I'm not sure if they are relics. Actually, Galapagos finches are relatively new, like they're recent in terms of colonization. So I'm not sure if they're relics in some way. I doubt that.
Robin, this is Ram. I've
Yeah.
since corrected my question, actually. I should have retracted it because I've done my own research. There is definitely a difference between relics is something that have sort of stayed on for a long period of time while their larger populations have disappeared. So
Yeah.
I certainly thought of Galapagos finches. Definitely, that's more of an evolution, a radiation type of thing as opposed to relics.
That's right.
Maybe the
Yeah.
Hudson in Peru, in Ecuador, the Hudson may be a better reference, I think, for a relic than the honey creeper. Or I'm sorry about that question. I've since
No
then
worries. No worries.
corrected myself.
Yeah. Also, another question from Ram is the green side of the tree established based on fossils. Does the establishment of the phylogeny tree always need the DNA analysis, DNA samples?
So I think there were relationships that were depicted earlier, but those were not based on genetic data. So that is what is used to be called cladistics, where relationships were examined based on shared traits. So you can build a similar tree, but based on shared traits. But when the traits are then, they are genes, that is when that's when the game changed because it became much more informative, and you could differentiate from convergent evolution. Yeah.
Yeah. I hope that's clear. And okay, so there is another question from Mayank. Can oceanic currents act as biogeographic barriers?
Yes, that is very, very true. In some cases, there are parts of uh, islands and things like that where there are these barriers. So definitely, it's a very important barrier actually.
Yeah. And the second question from Mayank, in the map of distribution of biogeographic pattern, shown interestingly, species of eastern origin are found to be common in western parts of India, and species with western origin are more common in eastern parts of India. Any specific reason for this?
Is that really true? Eastern part in western and the western part in eastern.
Yeah. So the
Uh,
species of eastern origin are found to be common in western parts of
no,
India.
that's not true.
Uh,
I think this is referring to one of the studies, right? Maybe
yes.
Umesh is a co-author in that. I don't remember, but this is the mammals, no?
Yes.
Kama et al. Yeah, no, that it just shows that there's kind of some dispersal colonization from east, and a lot of the eastern elements are in the northeast, and whereas from a lot of the western dry arid habitat ones are in the west. That's what that shows. If I have said that incorrectly in the talk, then uh, that's not right. I mean, what I just said is the correct one.
Yeah, thank you, Robin. I think we covered the questions from discussion forum, and there are no questions in the students' response sheet. So if you have any doubt, please. Okay. So one
There's
more
one in the
question.
chat
Yes. Yes.
that I can read. So this is
Yeah.
Godavari river as a paleo river. Basically, I mean, this paleo river idea, I mean, even Bharatapura in the Palghat Gap is considered a paleo river because there's an ancient geological fissure, and there is water in that. So sometimes it can be from what is that called? Old marine incursions also have happened from there. So you find marine content also deep inside. So that is what I think the reason why it's called as a paleo river. So as in it's existed for a very long time.
Hi, Robin. So there is a comment in the chat that, again from Mr. Ramamurthy, if there is time, I can comment on measurement system analysis.
In terms
Yes,
of
absolutely. Please do. Would love to learn something about measurement system analysis.
Okay. Yeah, I couldn't attend the session on March 14th because I was traveling. There was a discussion from you about measurements of angles and distances, right? And so my question at that time was, great, that's awesome. But and you talk about in the slide, you talk about the importance of precision in the measurement. That is, how important it is to have the errors to be very minimum. So my logical question was, how do we know, do ecologists use statistical measures for measurement system analysis? Well, you answered the question on March 14th, but so I just thought today, if we had time, I would comment on it. So in the automotive industry, and especially in the aerospace and automotive industry, there has been a practice for over 70 years now. It's been 30 years since I left the automotive. It's been there for 40 years prior to that. So now I estimate that to be about 70 years old. There is the statistical measures that we do before any product is shipped to a customer. It has got to be measured. Some dimensions have to be measured for tolerances, because those are very, very critical, especially in aerospace and automotive industry. Like it's any part that you make. There are well over 14,000 parts in an automotive. Every part has to be measured. But before a measurement is made and sent out as quality control, one goes through an elaborate process of what we call measurement system analysis. In very simple terms, if the measurement is a continuous measure, like distance, so then the actual measurement is it's very simple at a very high level. It's about bringing in multiple operators who are possibly measuring. In the case of ecology, you're training a younger person out there in the field, and you are the mentor. You're going there and you're trying to show this younger person to say, hey, here's how you measure the distance. Here's how you measure the angle. Those are continuous measures. Then essentially what you do is you bring in a few people and you set a standard for distances and angles. Let us say you mark a tree where you have a set distance and a set angle, and you measure it at different heights. Then whatever the mentor measures is considered as, let us say, a standard, because you know this business. Then essentially what it does is it does an ANOVA analysis to measure within operator variance and between operator variance. Then it also sees, if let us say that you have an angle measurement unit, let us say that you have a more sophisticated angle measurement unit, then it sees if there is a variation as respect to, is there an interaction between a specific gauge, we call it gauge in automotive, between the gauge and the person. Does he have problems in handling this particular gauge versus that particular gauge? So it's a very elaborate measurement system. It's ANOVA. Just within operator and between operator measurements for seeing where the variation comes from. And then the idea is, you spot where the variation is, and then you go ahead and take appropriate countermeasures to see those variations are minimum, and that means the errors are minimum. That is for if the measures are continuous. But what if you're measuring color, right? And you don't have, let us say, spectrographic measurements for wavelengths of color, for color. You are just saying standard, like an ordinal scale for color. Then you set a standard. Now it's an attribute measure. An ordinal measure, it's an attribute measure. It's not continuous. You can't use ANOVA. Then there is a much more sophisticated attribute-based analysis for measuring agreement. It's called attribute agreement analysis. We use that in color standards because I come from the automotive color industry, from the color part of automotive paint is my specialty. So we use attribute agreement analysis because if somebody is making a standard to say that, hey, this is the standard, this is as good as the standard, this is better, this is worse. So there's very well established statistical measures to do that. So if it's okay, I can put the actual references to the papers because there's are all very, very old. These are all very old standards. I can put them on the discussion board for everybody else. I just thought I would comment this. It's ANOVA analysis or its attribute analysis. In fact, I was just thinking when I show up in December, I could actually work with somebody to set up this process and a simple line transect type of measurement to actually bring in 2 or 3 people and actually do a demonstration of this. It's very easy to do. It's extremely easy to do. And analysis is very straightforward. I'll be more than happy to do it. Maybe Jubilee
Yes,
Gardens.
I mean, sorry. No, thank you that very detailed explanation, and also thank you for the very kind offer. That's in fact there are, I will comment earlier on the continuous variables, and I think it's useful to have this discussion because a lot of the things that we do in terms of training and then data collection and data curation actually have to do with dealing with these kinds of errors. And so when we're measuring continuous variables, whether it's like distance and angles, of course, there is a whole training component to that, and you get better with time, obviously. So somebody who's learning collecting data on a line transit or point counts is actually going to repeatedly measure angles, known angles and known distances until that inter-observer variation becomes acceptable, becomes small enough that it's not going to influence your results. So there's a lot of training that happens before somebody is going out and actually measuring distances and angles in the real world on a line concept. Now, uh, unfortunately, with line transits, of course, we can't repeatedly measure the angles of the angles and distances to the birds because of other things. You need to walk a line transit at a certain pace, so you can't, stop and do repeated measurements of the bush where the bird was sitting on, for example. That unfortunately not possible. But one of the things ecologists do is recognizing that people will become better over time at making these measurements is to discard some proportion of the initial data and not include it in the final analysis. So that's one way in which ecologists sort account for observers becoming better and more precise over time. More accurate and more precise. So, Amrita, since should we just wait for a few minutes in case somebody's typing something into the chat box?
Yeah,
Uh,
yeah, we can.
and if there are no more questions or comments, we can end the meeting.
Sure. I think we'll wait 5 more minutes. Is that okay?
Sure,
Yeah.
sure.
I think if there aren't any questions, we'll wind up the session. Thank you so much, everyone, for joining in, and thank you, Umesh. Yeah,
All
thank you.
right. Thank you so much for joining, and thank you, Amrita, for coordinating all of this. And we'll see you next week.
Yeah, thank you.